Hello, this is Edward Ceballero, and I will be covering today's current events project on cryptochrome. Peter On and myself put it together, but I will be narrating. Cryptochrome. I chose this molecule because I read several articles over the years about all the discoveries surrounding it and a lot of the mystery that surrounds it is what enticed me to choose this as a topic. So what is cryptochrome? Cryptochrome, also known as cry, is a class of flavor proteins that encompasses a blue light driven reaction cycle. Cryptochrome exists in a multitude of varieties. It's similar to photolase structurally. Although they are functionally different, they possess similar photoactive domains. However, more is known about photolase than cryptochrome. Cryptochrome is involved in the circadian clocks of plants and animals, and in the sensing of magnetic fields in a number of species. Discovery. Phototropism, specifically with blue light, was first documented by Charles Darwin in the 1800s. However, it was not until the 1980s that research began to identify the pigment responsible. In 1980, researchers discovered that the HY4 gene of the plant Arabidopsis thaliana was necessary for the plant's blue light sensitivity. Spurring a renewed curiosity, the gene was sequenced in 1993. By 1995, it became clear that the gene and its two human homologs were a new class of blue light photoreceptor, hypothesized to be circadian photopigments. Interaction in animals. Known as magnetoreception, the magnetic sense in birds is thought to be mediated by the blue light receptor protein, with potentially some interaction with the magnetite found in their beaks. Magnetotactic bacteria possess magnetite and are known to align to magnetic fields in a phenomenon known as magnetotaxis. The mechanism behind cry's magnetoreception is largely unclear. However, gene knockouts of cryptochrome have been proven to cause disorientation in Drosophila melanogaster, who are trained to respond to these magnetic fields. Cryptochrome in humans. Humans run on a 24-hour clock, also known as the circadian rhythm. It is synchronized to the rising and setting of the sun. Body systems are calibrated to the appearance and disappearance of light. Clock genes regulate our 24-hour clock. These genes include BMAL1, clock, period, and, of course, cryptochrome. This is an image kind of simplifying or illustrating how the circadian clock works in humans. It regulates body temperature, melatonin secretion, and various other metabolic processes. Cryptochrome in humans continued. Clock genes work together, like cogs in a mechanical watch, to control our daily, cyclical, physiological changes like temperature, blood pressure, and sleeping cycles. We tend to have the best cognitive functioning during the late morning. This is our clock genes at work. Normally, we get sleepy when it gets late. This is due to there being more melatonin released at this time. And this is just one example of our clock genes at work. Another example is jet lag. When we try to fast forward or rewind the natural 24-hour day, such as when we fly 7 time zones away, our circadian clocks don't let us off so easily. The genes and proteins need time to adjust. Jet lag is the feeling of our cells realigning to their new environment and the starting point of a new solar day. So what does cryptochrome do? Until now, scientists understood how clock genes worked collectively, but not individually. unlike animals, cryptochrome in humans has not been shown to aid in our magnetic orientation. We didn't know what exactly they did, what cryptochrome did, at least not in humans. However, new research by the University of North Carolina has recently uncovered the function of cryptochrome in human beings. The function of cryptochrome. Continue. At the start of the day, BAM1 and clock work together to start the circadian clock. They bind to other genes and turn them on, making them express proteins. They also bind to period and cryptochrome, turning them on to express proteins, which together suppress BMAL1 and clock. The suppression of BMAL1 and clock stops when period and cryptochrome's expressed proteins are degraded, at which point BMAL1 clock start completing the 24-hour cycle. The function of cryptochrome. To find what cryptochrome did by itself, scientist Chris Selby, PhD, created the first mutant cell that only had BMAL1 and clock. To be specific, there was no cryptochrome or period present. Period was put into this mutant cell and did nothing. Cryptochrome by itself didn't just suppress BMAL1 clock, but completely shut them down. In other words, the circadian clock was stopped. Period was then reintroduced to the shut down mutant cell. It was removed, the cryptochrome was removed, allowing the circadian clock to restart. So, essentially they isolated all these individual genes to find out exactly what cryptochrome did. To further simplify, BMAL1 clock are the start of the clock. Cryptochrome is what begins to signal the clock to come to a finish. Period is what gets rid of cryptochrome so that a new cycle can start. An inverse relationship exists between period-paired cryptochrome and clock concentrations. And this is a few illustrations of that inverse relationship, rather. Significance. Before, all we knew was that clock genes have something to do with our circadian rhythm. Now we are a step closer to better understanding the individual functions of each clock gene. We have a better understanding of how clock genes interact and how the entire clock works. Figuring out the function of cryptochrome was a huge step in this. Furthermore, on the significance, this has significance in research focused on treating jet lag, cancer, target cancer cells circadian clock with cancer drugs is one usage of this. Seasonal affective disorder, mood disorder, metabolism disorders, sleep disorder, bipolar disorder, and pregnancy disorders. Alright, here's some future applications. Perhaps there are many more functions of each individual clock gene still to be discovered. Suppress or amplify certain specific clock gene expressions. Turn off certain specific unwanted clock gene expressions. What if we were able to alter a circadian rhythm and flip what the body perceives as day and night? This would be great for people who work at night and sleep during the day. What if we were able to take our body's peak cognitive performance time of the day and make it last all day? And these are my sources. And that's all. Alright, so that covers my presentation. If you have any questions regarding the topic, please post them in the comments. And thank you very much.